I like cooking, but let me tell you what I don't like. I don't like getting cut. An unfortunate side effect with working with knives in the kitchen is that eventually you're inevitably going to slip and accidentally hurt yourself. Today that exact thing happened to me and I missed a slice and I ended up bleeding a bit. But luckily that one slip isn't a death sentence for me. Because I'm human, or at least as far as you're aware, my body is able to build up its defences once again and repair those skin cells that have been damaged. But the question still stands, why is it that my skin is able to repair itself? And why is it that all of the skin cells on my body are practically identical to each other? Let me explain. Before we get into any details about exactly what is happening in the process of this, I think it's first important to understand the exact molecules that are being used here. The most obvious of which is DNA. I'm sure you've all heard of DNA, whether that's just in passing or you've actually seen something about it. But the important thing to know is how DNA is actually structured in biology. DNA stands for deoxyribose nucleic acid. DNA is found in all living things and is a vital molecule for genetic code. As far as humans go, it codes for everything, including our eye colour, our hair colour, how big and strong we are. That is all stored in the DNA. And without this structure, much of our life would not be able to exist. The same importance can be laid on RNA, yet for humans at least, this compound is not nearly as prevalent, but we would definitely not be where we are today if RNA did not exist. RNA is much like DNA, however it has a lot shorter of a structure compared to the double helix of DNA that stretches for miles and miles and miles. RNA is mainly made up of small segments of a single helix. Speaking of stretching for miles and miles and miles, this type of macromolecule can be described as a polymer. Both RNA and DNA fall under this category. RNA has the ability to connect directly to DNA and translate data from the DNA strand to the RNA strand, and we'll learn about that next time. But for now, all you need to know is that there are 4 bases in each of these molecules. Bases is just the opposite to acid, as I'm sure you know, and we'll come to learn why that's important in just a sec. But for now, the bases make up of adenine, thymine, cytosine, and guanine for DNA. RNA is exactly the same except from one base, which is changed for uracil, and that base is thymine. We always refer to these in biology when we can by their initial letter, so A, T, G, C, and U on RNA. These bases form what are called complementary pairs. Much like enzymes, one of them will be able to fit to another one, and that's because of the intermolecular forces that occur between them, namely hydrogen bonds. For the A base, we have a T. For the G base, we have a C. And if you remember what I've just said, uracil can replace that T when it's referring to RNA. This allows RNA to stick to DNA by creating a complementary base pair on its spine. Speaking of the spine, it's important to understand what the backbone of this structure is, because at the minute we're just talking about bases, but DNA is a lot more than that, and if you remember the name, they're called nucleic acids, not just bases. When we're discussing DNA, what we're talking about is the structure of a phosphate group at the head, connected directly to a pentose sugar. And when we're describing sugars, if you recall the saccharides video, we talk about sugars that have different numbers, and we simply put the prefix followed by an "ose." So in this case, it would be pentose for the 5-carbon sugar that we're creating. We refer to the long chain of molecules that make up this DNA strand as a sugar-phosphate backbone. The sugar comes from the pentose and the phosphate comes from the phosphate group. A simple thing we have to remember. The pentose also has a base attached to it, and we refer to this base as a nitrogenous base. Nitrogenous base simply means one that has nitrogen in it, and in the case of A, T, G and C, they all have nitrogen within their compound, and this is exactly where they're going to connect. If you remember the acronym of DNA, you might be thinking, what deoxyribose means? Well, in this case, deoxyribose is that pentose sugar that we've just discussed. And in RNA, this is exactly the same structure, only that RNA will have a lot shorter of a chain than the DNA strand. Now here's a question. How will we form these DNA strands and RNA strands? Condensation? No way. Again, condensation reaction appears in this formation of this compound. Simple enough to remember. In this situation, what will happen is the phosphate group and the pentose sugar will connect to each other, releasing a water molecule. Now finally, one of the smaller topics, yet it's still important to understand. Historically, we were quite confused as to what stored our data for all of our genetic components. And when this idea of this simple, very basic looking, pun not intended, double helix contained all of the genetic information for our bodies, people were naturally skeptical. So it was thought for a long time that DNA didn't actually contain our genetic code until science experiments actually proved it. One of those science experiments we'll come on to next episode. Until then, I'll see you next time, and hopefully by that time my finger will have healed.